Hello children, hope you are working very hard. Shall I tell you, there is one more process occurring in your body who is working even more harder, all times, without any break. The one which is there on the board, respiration. So today we are going to discuss in detail the most hard-working process in your body, without any break, occur relentlessly. And that process is said to be respiration. So therefore, we are going to study this process without wasting any time. What is the definition of respiration? It is breakdown. It is a breakdown process. It is a breakdown process or oxidation process of organic compounds. This organic compound can be a fat, can be a protein, or can be a carbohydrate. If it is completely breaks into CO2 water, or it may even undergo partial breakdown, producing alcohol and acid. And that process is said to be anaerobic. All times it occurs. In all living cells it occurs. Stored energy is converted to ATP. The glucose, the CC bonds, they are converted to ATP. So that is one more type, very important. It is amphibolic process. You know what is amphibolic? Not only it is catabolic. Some of the respiratory intermediates like PGL, it can produce glycerol. Acetyl coenzyme A, it can produce fatty acid. So it means that not only it is catabolic but also anabolic, put together amphibolic. Where it occurs? It occurs in cytoplasm and even in mitochondria, provided if it is aerobic respiration. Link reaction, Krebs cycle and ETS occur in mitochondria. What is the difference between combustion and respiration? In combustion, energy is released in a single step, whereas in respiration, energy is released in a stepwise manner so that ATP can be synthesized. That's a big difference between combustion and respiration. All right, in many steps. In respiration, single step here and many steps here. All right, then energy lost due to the breakdown of CC bonds is mostly in the form of heat. That's a very good question. When the CC bonds of glucose is broken, children, approximately I'll tell you, totally if it breaks into CO2 and water, approximately 700 kilocalories of energy is liberated, out of which 400 kilocalories is going to maintain heat, is lost in the form of heat energy, maintain your body temperature. 700 minus 400, 300. The remaining 300 kilocalories is converted to ATP and stored in your body as energy currency. Approximately to synthesize one ATP, we require 7 kilocalories. So we have 300. 300 by 7, approximately 40 ATP are produced for 1 molecule of glucose. Is it okay? Then say for example, if I want to run, I require some energy. ATP breaks and give me the energy. First, I use energy to synthesize ATP. When I want energy, ATP break to liberate that. All right. Then ATP is a nucleotide. It contains ribose sugar, adenine, and phosphate, ribose sugar. Respiratory organs are seen in animals. Lungs, gills, and moist skin are the respiratory organs in animals. What about plants? Stomata and lenticels are respiratory organs. No, they are not organs. They are apertures. How can apertures be organs? They are not respiratory organs. They aid in exchange of gases, but they are not respiratory organs. Equation, this compound is losing hydrogens, oxidation. This is gaining hydrogens, reduction. Glucose is oxidized, oxygen is reduced. What is the difference between breathing and respiration? I was doing the job of breathing. Taking in of oxygen and liberating CO2 is breathing. All right, what is respiration? Breakdown of glucose in your body cells into carbon dioxide, water, and energy is respiration. So breathing is prerequisite for respiration to occur. Okay, understand that. I explained when you take oxygen, it enters into your lungs, alveoli, blood capillaries. Then this oxygenated blood is transported to heart. Heart is going to pump the oxygenated blood to all parts of your body. In your body cells, glucose is present. In the presence of oxygen, it breaks into CO2, water, and energy. That process is said to be respiration. What are respiratory substrates? Carbohydrates, fats, proteins, and organic acids. Look at it. Fats, 1 gram of fats is going to give approximately 9 kilocalories. Be careful, children, when you eat fat. More fat if you consume, and if it doesn't do enough work to break it, then the additional amount of energy which is liberated from 1 gram of fat, oxidation will be stored in the form of adipose tissue in your body. So therefore, when you consume more fat, do more exercise. Otherwise, consume less fat. Now, respiratory organs are absent in plants. Why? 3 reasons. The metabolic rate in plants is less. Plants do not present greater demand of gaseous exchange. There is no need of maintaining body temperature in plants. Plants doesn't run, plant doesn't think. All right. So therefore, the amount of metabolism in plants is far less when compared to animal. Therefore, there are no respiratory organs. Then, every plant part can take care of their own oxygen needs. Roots can take oxygen by diffusion, stem can take by diffusion, leaves can take by diffusion. Stem will never ask leaf, leaf will never ask root for oxygen. That is one more reason. Then, even in large bulky plants, intercellular spaces are present. More oxygen can enter into that and can stay in the intercellular spaces, which can help in exchange of gases. So therefore, even if a plant cell is deep-seated, no problem. Intercellular spaces have enough oxygen. It can grab that oxygen from them. So that is one more reason. Large volumes of gaseous exchange occur during photosynthesis. Come on, children, this you know it. In photosynthesis, plants take CO2 and liberate oxygen. And that oxygen is taken by cells for respiration and liberate CO2. So therefore, large. Every living cell is very close to the atmosphere in leaf because it is dorsiventally flattened. Loose packaging of parenchyma is seen in root, stem and leaf for exchange of gases. These words are very important. Obligate aerobes, compulsory aerobes. You cannot survive in the absence of oxygen. Can you? No. So you are example for that. Facultative anaerobes. Sometimes anaerobes. Ante mostly they are aerobes, but can survive even in the absence of oxygen. So mycoplasma is an example for that. E. coli, yeast, very important, facultative anaerobe. Obligate anaerobes, compulsory anaerobes. Food poisoning occur when you tightly pack your foodstuffs because this bacteria will be very active. Clostridium, it's an obligate anaerobe. Facultative anaerobes, sometimes aerobes, means mostly anaerobes, sometimes aerobes. Example, green sulfur bacteria. Now children, we'll get into a phase called glycolysis. The very first phase, all living organisms retain the enzyme machinery to partially oxidize glucose. This partial oxidation of glucose is glycolysis. We derive from Greek word, it occurs in cytoplasm, discovered by Emden, Meyerhoff and Peranus. Common name, cytoplasmic respiration or EMP pathway. Oxygen is not needed, CO2 is not produced. It is partial oxidation of glucose. It is universally seen in all organisms. Anaerobes, it is the only process of respiration. ATP is first invested and later produced. Cell organelle is not involved. And what are the substrates for glycolysis? The substrates could be glucose or fructose, any of them. Not sucrose. First, sucrose breaks in the presence of invertase. Is the breakdown of sucrose can be taken as glycolysis reaction? No. All right. So glucose, it can be converted to glucose 6-phosphate, or fructose, it can be converted to fructose 6-phosphate and fructose 1,6-diphosphate. Let me write the reactions of glycolysis. We are not going to take much time because you know all the reactions. Today we'll try to find some ways to remember it more easily. All right, first reaction. Look here. Glucose is converted to glucose 6-phosphate. Then it is converted to fructose 6-phosphate. Then it is converted to fructose 1,6-diphosphate. Then it is going to break into DHAP and PGAL. And DHAP converts to PGL. I'll stop here. Now look what is happening here. Please concentrate what is happening here. Look here. Look at the substrate product, children. You need not know anything about the chemistry. A substrate product. Glucose is converted to glucose 6-phosphate. Means on this glucose, on the 6th carbon, there is a phosphate. It means that you have to add ATP. This is phosphorylation. ATP is donating one phosphate. That is going to the 6th carbon. Already you know glucose and fructose are isomers. Therefore, this reaction is isomerization. Aldo sugar, keto sugar. Then Fructose 6-phosphate. On the 6th carbon there is phosphate. Fructose 1,6-diphosphate. On this, even on the first carbon there is phosphate. Means somebody is donating phosphate. Again, ATP. 6 carbon is breaking down to 2,3 carbon compounds. This is cleavage. Enzyme name is aldolase. And the DHAP is converted to PGL. This is aldo sugar. This is keto sugar. Sorry, this is aldo sugar. Both of them are functional isomers. So, phosphorylation, isomerization, phosphorylation, cleavage, and again isomerization. So easy to remember the first 5 reactions. Isn't it, children? Kinase, isomerase, kinase, aldolase, isomerase. How many PGL molecules are there with me now? 2. Why 2? 1 from here, 1 from here. 2 molecules of PGL. First 5 reactions, we took a small break and we understood these reactions. They're looking very fine now. All right, now we go to the next 5 reactions. What are the next 5 reactions? Look here. 2 molecules of PGAL converted to 2 molecules of 1,3-dipga. This is converted to 2 molecules of 3-PGA. Then this is converted to 2 molecules of PEP. I'm sorry, 2 molecules of 2-PGA. Then 2 molecules of PEP. And then 2 molecules of pyruvic acid. I repeat again. PGL to 1,3-dipga, 1,3-dipga to 3-PGA, 3-PGA to 2-PGA. All right, 2-PGA to PEP, pyruvic acid. These are the reactions. Let us see. Let us see what type of reactions they are. Please concentrate. We are up to here. The same 2-PGL I'm taking here. Let us look. Just look. Little bit of chemistry used, children. Aldehyde, acid. When can aldehyde become acid? On oxidation. Oxidation means loss of hydrogen. Finished. 2 hydrogen molecules lost. Accepted by 2 NAD to form 2 NADH2. And I want to tell you, each hydrogen atom is 1 redox equivalent. 2 to the 4, 4 hydrogen atoms means 4 redox equivalents are lost from 2 PGL. If they ask you for 1 PGL, how many redox equivalents? Your answer should be 2. And what I produced is 2 molecules of NADH2. NAD is a coenzyme. I'm adding hydrogens to it. Reduced coenzyme. Reduced coenzymes are produced. Next, 1,3-dipga is converted to 3-PGA. First carbon, third carbon, there is phosphate. Only on the third carbon there is phosphate. Phosphate on the first carbon is lost. Accepted by ADP to form ATP. First time ATP is formed. This is substrate level phosphorylation. Yes or no? Yes. 3-PGA, 2-PGA. Phosphate on the third carbon is shifted to the second carbon on the same molecule. Intramolecular shift. This is positional isomerism, isomerase. 2-PGA to PP. Water is lost. Dehydration. 2 molecules of PP to pyruvic acid. Phosphoenol pyruvic, just pyruvic. Finished. Phosphate is lost to ATP. Is it difficult? No, children, it is not difficult. Because aldehyde acid oxidation, 1,3-dipga, 3-PGA. Dephosphorylation or substrate level phosphorylation, both are right. First, phosphate is lost from this. Then that phosphate is accepted by ADP to form ATP. Then intramolecular shift, isomerization. Then this is dehydration. Then this is dephosphorylation. Shall I tell you the easy way to remember? Look here. This is from here onwards 2 molecules. From here onwards 2 molecules. I want to use a 2-letter word. You know what the 2-letter word? DE. DE. Dehydrogenation. Yes. Dephosphorylation. Yes. We'll take a small break. Then dehydration. Yes. Then dephosphorylation. This is interesting. All right, look here. One more way of saying. Dehydrogenation is followed by dephosphorylation. Dehydration is followed by dephosphorylation. Easy way. Dehydrogenation followed by dephosphorylation. Small break, isomerization. Dehydration followed by dephosphorylation. This is looking fine. This is looking fine. All right, and these are the reactions which you have to remember. 4 redox equivalents lost, that point is important. This is the first energy yielding step because ATP is formed. This word substrate level phosphorylation is important. Why you use the word dephosphorylation and substrate level phosphorylation? I already told you. The substrate is losing a phosphate, dephosphorylation. This is accepted by ADP to form ATP, substrate level phosphorylation. Easy way to remember. All these 10 reactions occur in cytoplasm. Let us see how many carbon compound it is. 6, 6, 6, 6, 3, 3. These are all sugars. All right. Then non-sugar 3. This is not a sugar 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 3. Glycolysis reactions finished, and we have completed nicely and successfully. And what are the points which I have written with regard to glycolysis? Let us see, children. Let us see what I have written. I have written the reactions. Glucose, glucose 6-phosphate, fructose, 1,6-PGL, DHAP, 1,3-dipga, 3-PGA, 2-PGA, PEP, pyruvic acid. Correct. What about fructose? Fructose is converted to fructose 6-phosphate, 1,6-diphosphate, PGL, DHAP, 1,3-dipga, 3-PGA, 2-PGA, pyruvic acid. Correct. Don't think that fructose become glucose. No need. Fructose can directly become fructose 6-phosphate. Like here. Fructose can directly become fructose 6-phosphate and continue like this. These are the enzymes of glycolysis, you know it. Gross ATP, 4 ATP formed. Where? 2 molecules of PEP to 2 molecules of pyruvic acid, 2 ATP. Previously, 2 molecules of 1,3-dipga to 3-PGA, 2 ATP, 4 ATP. Net is 2. Gross means total. Net means gain. 2 are utilized. First reaction, third reaction. Therefore, 4 minus 2, the net is 2. 2 NAD, H plus are produced. Correct. 2 water produced. 2 molecules of pyruvic acid produced. Metabolic fate of pyruvic acid depends upon the cellular need. We'll discuss that. Number of ways, 3. 6 carbon, this 3 carbon sugars. These are 3 carbon non-sugars. This is important. Substrate level phosphorylation, 1,3-dipga to 3-PGA, PEP. This is again important. Oxidation, PGL to 1,3-dipga. For 1 PGL, the number of redox equivalents are 2 because there is 1 hydrogen molecule lost, 2 hydrogen atom. For 2 PGL, 4. Very important. Isomerization, glucose to fructose, DHAP to PGL, 3-PGA to 2-PGA. Phosphorylation, glucose to glucose, fructose to fructose. Even PGL, inorganic phosphates are added, children, to form 1,3-dipga. Even this is phosphorylation. Dehydration, you know it. Isomerizations 3, dephosphorylations 3, substrate level phosphorylations 2, oxidation 1, cleavage 1, dehydration, remember the enolase, and for cleavage, remember aldolase. Kinase mediated steps 4, isomerization involving intramolecular shift 1, mutase. In glycolysis, ATP is first utilized and then produced. ATP produced is 4, utilized is 2, net gain is 2, no organelle involved, reduced coenzymes get No. Reduced coenzymes are produced. They don't get oxidized, okay? Are produced. Magnesium is needed for kinase mediated steps. What is the coenzyme? NAD. What is reduced coenzyme? NADH2. Are they produced? Yes. All right. Magnesium is needed for kinase mediated phosphofructokinase, allosteric, not important. Floating respiration, carbohydrate acting as not important. Protoplasmic proteins, not important. Chalo, we completed glycolysis. Now we get into the next phase called fermentation. What will happen to pyruvic acid? 3 things will happen, children. All right, before we get into these words, what will happen to pyruvic acid, I'll tell you. Listen carefully. This is pyruvic acid. This is pyruvic acid. Okay, if I'm adding NADH2, listen here. 2-keto-propanoic acid is pyruvic acid. 1 hydrogen goes and joins here, other hydrogen goes and joins here. Then it becomes dash CH3, COH, H, and COOH. And what is this called? This is called lactic acid. This is called lactic acid. It occurs in your muscle cells in oxygen deprivation. All right. Lactic acid accumulation results in fatigue. You heard about that. All right. This is the lactic acid produced. Sometimes the curd is very sour. The reason, more acid. Which acid? Lactic acid. What is the bacterium? Lactobacillus. So the pyruvic acid is getting reduced. Reduced? Yes. Why? Because it's getting hydrogens. From where? NADH2. Where it is produced? In glycolysis. This is lactic acid fermentation. Then what the next type of reaction, children? Next type of reaction is alcoholic fermentation. Look what happens in alcoholic fermentation. Again, I take pyruvic acid. This time I remove CO2. When I remove CO2, it becomes acetaldehyde, so easy to remember. Now I'm going to add NADH2. Then what will happen, children? One hydrogen will go and join here. Other will go and join here. Then what is this called? Ethyl alcohol. Finished. First decarboxylation followed by reduction. You may ask me, is reduction common in both? It's common in both. Lactic acid fermentation. Why? Only on reduction. All right. Means for reduction to occur, NADH2 has to lose hydrogen, oxidation. Only when it loses hydrogen, this NAD can run into glycolysis and allow the continuation of glycolysis, because NAD is again needed for PGL. This is fermentation, children. Let us see what I have written. Two types of fermentation. First, alcoholic. Pyruvic acid undergo decarboxylation. Acetaldehyde is formed. Acetaldehyde undergo reduction to produce ethyl alcohol. NADH2 should undergo oxidation so that it can again participate in glycolysis. It occurs in yeast. Net ATP is 4. Net ATP is 2 because 4 produced, 2 utilized. Net NADH2 is 0. Why? 2 produced in glycolysis, utilized in the conversion of acetaldehyde to ethyl alcohol. Net CO2 will be 2. Yes or no? Reactions, decarboxylation and reduction. Then coming to lactic acid fermentation, I told you, pyruvic acid plus NADH2 give rise to lactic acid plus NAD. All right. Occurs in lactobacillus muscle cells. It's the reduction of to produce lactic acid, pyruvic acid. Net ATP is 2. Net NADH2 is 0. Net CO2 is 0. There is no CO2 formed. Net CO2 is 0. There is no CO2 formed because pyruvic acid is directly forming lactic acid. It is not forming acetaldehyde. That is very easy one. The same thing I have written here. You can go through this, children, by pausing the video. Now we go to the next one, that is aerobic respiration. What is aerobic respiration? Pyruvic acid has 3 ways. A, it can form lactic acid. B, it can form ethyl alcohol in yeast. 3, it have a choice of getting into mitochondria and participating in aerobic respiration. So after getting rid of fermentation successfully, now we get into aerobic respiration. What will happen to 2 molecules of pyruvic acid if it is in aerobic respiration? There are 2 crucial events in aerobic respiration, children. One, we completely oxidize pyruvic acid, means we remove all the hydrogens from pyruvic acid. Then what we'll do with these hydrogens? Add to NAD to form NADH2. Add to FAD to form FADH2. Finished. When we remove all the hydrogens from 2 molecules of pyruvic acid, then naturally the pyruvic acid becomes CO2 because you're removing hydrogens. So there is a loss of CO2. So this is the meaning of complete oxidation of pyruvic acid. Means you are going to completely, in link reaction and Krebs cycle, in these 2 phases, I'm going to remove all the hydrogen atoms from pyruvic acid, give to NAD and FAD such that they form NADH2 and FADH2, and that leaves only CO2. Yes or no? Then next step in aerobic respiration, electron transport to oxygen to produce ATP. What is this? I told you NADH2 and FADH2 are formed here. Is it not? What I do with that NADH2? I forcibly remove hydrogens from NADH2 and forcibly remove what is called hydrogens from FADH2. Then what I do? I'll give it to oxygen, and that electron transport will release energy and produce ATP. That is said to be oxidative phosphorylation. It occurs on cristae. So the next step is electron transport to oxygen. So first from pyruvic acid, I remove hydrogen. Example, link reaction and Krebs cycle. What is formed? NADH2 and FADH2. Then what I do? I take this NADH2 and FADH2 to electron transport system. I throw the hydrogens from them to oxygen. Oxygen is going to grab those hydrogens. In this entire electron transport chain, ATP is produced. That is called oxidative phosphorylation. All right. Now, where is complete oxidation of pyruvic acid occur? I told you. Complete breakdown of pyruvic acid occur. It occurs in mitochondrial matrix. 3 molecules of CO2 are formed. All right. Pathway. For 1 molecule of pyruvic acid, 3 molecules of CO2. For 2 molecules of pyruvic acid, come on, I told you pyruvic acid is a 3 carbon compound. When it undergo complete oxidation, so the number of carbons will be, number of CO2 will be 3. For 2 molecules, it is 6. All right. It occurs in mitochondrial matrix. And the 2 pathways involved is link reaction and Krebs cycle. Then I told you what we do in electron transport system. This NADH2 and FADH2, we give it to oxygen, final electron acceptor. In this process, we produce ATP, and that is said to be oxidative phosphorylation. Now we go back to the first step. What is the first step I told you? The first step is this, complete oxidation of pyruvic acid. In how many sub-steps it occurs? 2, link reaction and Krebs cycle. Now we go to the next one, that is link reaction. All right, what is link reaction? Some of you got a doubt. Again, I go back, children. This is pyruvic acid, 2-ketopyropanoic acid. In lactic acid fermentation, I added hydrogen HH, so then become lactic acid. In alcoholic fermentation, I removed the CO2. I'm sorry, acetaldehyde. This acetaldehyde, you are adding hydrogens to form methyl alcohol. Then what I do in aerobic respiration, listen carefully. I remove the CO2 first. Just like in alcoholic fermentation, I remove the CO2 first. What it forms? Acetaldehyde. Then you may ask me, are you adding hydrogens to acetaldehyde and form methyl alcohol? No. I not add hydrogens, I remove these hydrogens. And I give it to NAD to form NADH2. I already told you, complete oxidation of pyruvic acid, it results in the loss of hydrogen from pyruvic acid, accepted by NAD to form NADH2. The hydrogens are lost, acted by NAD to form NADH2. But the valency of carbon should be 4, na? But here the valency of carbon is only 3. If you remove all this, 1, 2, 3, how can I make 4? I add coenzyme A. Then it forms acetyl coenzyme A. Finished. How many carbons? 1, 2. This is not carbon. Okay, be careful. 2 carbon. And this 2 carbon compound is acetyl coenzyme. So what did we do? First I removed CO2, then I removed hydrogen, then I added coenzyme A. Decarboxylation, oxidation and condensation. All right. What did I write here? Link reaction. Matrix, yes. Oxidation means loss of hydrogens, yes. Decarboxylation, yes. Enzyme pyruvic dehydrogenase complex, remember that. Magnesium is needed, very important. Not only magnesium, we require NAD, FAD, TPP and coenzyme A. Product, it's a connecting link, 2 carbon compound between glycolysis Krebs cycle, very important. Number of NADH2 formed is 2 because we have got 2 molecules of pyruvic acid. Number of CO2 formed is 2. And acetyl-CoA is a 2 carbon connecting link. We already discussed. First we removed CO2, then we removed hydrogen. In place of all this, I added coenzyme A, 2 carbon acetyl coenzyme A. So easy to remember, absolutely no problem. Then we go to Krebs cycle. What is Krebs cycle? So we have to start from acetyl-CoA. We have to break this acetyl-CoA into CO2 and water. That is Krebs cycle. It occurs in matrix. Scientist names is Krebs. Common name TCA cycle or organic acid cycle. Process, it is the complete breakdown of acetyl-CoA into CO2 and water. Complete breakdown of acetyl-CoA into CO2 and water. Be careful. Acetyl-CoA, not pyruvic acid. And I have told first step, last step, all these reactions in fact are known to you. Shall I make it first, write it in the form of a table? Then we can go through all these reactions very fast. Let us not waste much of our time, children. We'll run through this table first and then we'll be writing these reactions fast. All right, so first we'll write the table here. All right, now look at the table here. Very simple. 2 molecules of acetyl coenzyme A. This is 2 carbon compound. It is combining with 2 molecules of oxaloacetic acid. This is 4 carbon. Okay, 4 carbon, 4 plus 2. If you take 1 molecule, 4 plus 2, 6, and it forms citric acid. Citric acid. This is a 6 carbon compound. So 2 molecules of water is also used. And this reaction is called condensation. Why condensation? 4 carbon, 2 carbon, 4 carbon, 2 carbon, combining to form 6 carbon. Enzyme name citric synthetase. Okay. Chalo, what the next one? Citric acid is going to form its isomer. And its isomer, 2 molecules, I should not forget. All right, to form 2 molecules of isocitric acid. Nothing to talk about this reaction. Isomers are functional. So isomers are having same molecular weight, molecular formula. There should not be any bisubstrate and byproduct. Easy way to remember. Next two reactions, shall I tell you? Very simple. Go back to link reaction. What is the link reaction? Oxidative decarboxylation. Agreed? Same way. The next two reactions are oxidative decarboxylation reactions. Two molecules of isocitric acid forms two molecules of alpha ketoglutaric acid. And two molecules of alpha ketoglutaric acid forms two molecules of succinyl coenzyme A. Succinyl coenzyme A. All right. So these are the two these are the two oxidative decarboxylation reactions. Let me write with a different pen. Oxidative decarboxylation means there is loss of hydrogen 2NADH2. There is also a loss of carbon 2CO2. Again, there is a loss of hydrogen accepted by NAD and there is a loss of carbon. This is very simple. This is turning out to be very easy to remember. No problem at all. Two molecules of first reaction is very simple. 4 + 2 6. Then this is isomerization. Then I told you the easy way to remember. Go back to link reaction, I said. It is oxidative decarboxylation. Same. Oxidative decarboxylation, oxidative decarboxylation. This is actually a 5 carbon. Naturally, children. This is 6 carbon. Why 6 become 5? Because there's a loss of CO2. For two molecules, 2 CO2. Then 5, this become 4 carbon. Why? Because 5 carbon by losing 1 CO2 become 4 because there are 2 molecules, 2 CO2. Easy way to remember. No problem. First reaction is condensation, then isomerization, oxidative decarboxylation, oxidative decarboxylation. What should I do next? What should I do next? Look here, look here. This and this joining. Now what I do is I'm going in opposite direction, means this is breakdown and forms 2 molecules of succinic acid. This is breakdown because I'm moving in opposite direction. This is joining, this is breakdown. Agreed? What will happen if there is a breakdown? Coenzyme A is knocked out. Then what happens? Energy get liberated. Then what happens? That energy couples ADP and inorganic phosphate to form ATP. Can I call this as substrate level phosphorylation? Yes. Is substrate donating any phosphate from its pocket? No. Then why? Donating energy. Ultimately, it is helping in ATP synthesis. Yes. Therefore, this is a substrate level phosphorylation reaction. First GTP is formed, then ATP is formed. So 2 molecules of ATP is formed. This is a substrate level phosphorylation step. All right. Very important step. How many substrate level phosphorylation steps in entire aerobic respiration? 3. 1,3-dipga to 3-PGA. 1. PEP to pyruvic acid. 2. Succinyl-CoA to succinic acid. 3. Very important. Then what will happen to succinic acid is converted to 2 molecules of fumaric acid, then 2 molecules of malic acid and 2 molecules of oxaloacetic acid. Listen children, just listen here, listen here. On this side, if there is anything which is worth remembering, oxidative decarboxylation, isn't it? Because this is isomerization, nothing special about this. Oxidative decarboxylation. What about this side? This side, I want to use a short word, oxidation. I don't want to say decarboxylation, oxidation. So it means that there is loss of hydrogens here accepted by FAD to form FADH2. This is oxidation. There is a loss of hydrogens here accepted by NAD to form NADH2. And this is hydration. I'm adding water. Look here. This is such a, a very interesting thing I'll tell you. This is a very tasteless reaction. Same way, this is a tasteless reaction. You may ask, what is this tasteless? Not useful for any further utility, children, in electron transport system. This reaction product has no utility. Tasteless. On this side one, on this side one. Now, next one. Oxidative decarboxylation. Here, oxidation. 2 oxidative decarboxylations, 2 oxidations. Easy way to remember. So easy, just a coincidence of some words. Then this is joining and this is breakdown, cleavage. 2 minutes back you said it is substrate level phosphorylation. I said, which is right, both are right. First it breaks, liberate energy, and then synthesize ATP. Joining, breakdown, oxidative decarboxylation steps 2, oxidation steps 2. Did you follow what I said? Yes. So that's about the most important phase, children, that is Krebs cycle. So count how many NADH2 form. Very easy to remember. Very easy to remember. We have drawn 2 lines, 2 lines. This is oxidative decarboxylation. This is also oxidation. So therefore 2 NADH2, 2 NADH2, 2 NADH2 and 2 FADH2. So total 6 NADH2, provided if you say 1 molecule of glucose. If they ask you how many NADH2 are formed in Krebs cycle for 1 molecule of glucose, 2, 2, 2, 6. And how many FADH2 in Krebs cycle for 1 molecule of glucose, 2. But if they ask you how many NADH2 are formed in Krebs cycle, 1 Calvin cycle means we took 1 CO2 na. Krebs cycle means we should take it as 1 turn. So therefore everything is 1. So therefore 1, 1, 1. The number of NADH2 formed in Krebs cycle will be 3. Number of FADH2 formed in Krebs cycle will be 1. Number of ATP formed in Krebs cycle will be 1. Number of ATP formed in Krebs cycle for 1 molecule of glucose, there will be 2 turns, therefore 2. Number of NADH2 for 1 molecule of glucose, 2, 2, 2, 6. Number of FADH2 is 2. Be careful about the number of CO2 formed in Krebs cycle for 1 molecule of glucose, 4. Number of CO2 formed in Krebs cycle, yes, you told correctly, 2. 1, 1. Why 1? Because everything is 1 molecule. 1 CO2 here, 1 CO2 here. Master, you told nicely all the important points. All right, let us see what did I write with regard to Krebs cycle without wasting any time. Did I waste much time? I think I'm doing it. All right. 6 carbon compounds, citric, isocitric, oxalosuccinic. We did not discuss about isocitric. So citric and oxalosuccinic. So I think we did not discuss about oxalosuccinic, citric and isocitric. These two are tricarboxylic acids. And they get tricarboxylic acid pathway. This is 5 carbon and I'll change the pen. All right. This is 4 carbon compounds. Very easy one. First reaction, condensation, see there. Next reaction is isomerization, citric to isocitric. Next reaction, I told you, is oxidative decarboxylation, isocitric to alpha ketoglutaric. Remember, this is a 5 carbon compound. Next reaction is also oxidative decarboxylation. 5 becomes 4. Next reaction is important. Succinyl-CoA is becoming succinic acid. It is not only cleavage but also substrate level phosphorylation. Next reaction is oxidation. Succinic acid to fumaric acid. In enzymes chapter you studied malonate act as competitive inhibitor of this enzyme. And this is the only enzyme of Krebs cycle. Instead of present in matrix attached to membrane. Hydration, 2 molecules of fumaric acid becomes malic acid. Oxidation, malic acid becomes oxaloacetic acid. Again I repeat, oxidative phosphorylation, oxidative phosphorylation. So CO2 is also lost, CO2. And this is oxidation 2 NADH2, oxidation 2 FADH2 and substrate level phosphorylation 2 ATP. This is for 1 molecule of glucose. 6 NADH2, 2 FADH2 and 2 ATP. But it asks you in one Krebs cycle. So 1 NADH2, 1 NADH2, 1 NADH2, 1 FADH2 and 1 ATP. So in Krebs cycle, how many NADH2? 3. How many FADH2? 1. How many ATP? 1. I think I'm repeating many times. You are very good students, so you know all this. Products for 1 molecule of glucose. 1 molecule of glucose in Krebs cycle. CO2 4, NADH2 6, FADH2 2, ATP 2. Nice. Product in 1 turn or in 1 Krebs cycle. CO2 will be 2, NADH2 will be 3. All right, FADH2 is 1, ATP is 1. Product in link reaction and Krebs cycle put together for 1 molecule of glucose. Link reaction, there are 2 CO2, Krebs cycle 4, therefore 6. Link reaction, there are 2 NADH2 and Krebs cycle 6, therefore 8. In link reaction 0 in Krebs cycle for 1 molecule of glucose. In link reaction 0 ATP and 2 by substrate level phosphorylation 2. Oxidation steps are 4, decarboxylation steps are 2, substrate level phosphorylation 1, isomerization 1. Krebs cycle enzyme catalyzing dehydration and hydration is aconitase. Citric acid forms cis-aconitic acid. In some organisms, cis-aconitic acid again undergo hydration to form isocitric acid. Catalyzing hydration only is fumarase. This is very important. Succinic dehydrogenase attached to membrane. All right. Malonate is inhibitor. So this is something which we have written already, children. I don't think there is any need for me to go through all these reactions. That's it. That's about Krebs cycle. Now it is time for us to talk about what is called the last step of this entire respiration chapter, that is electron transport system. And I told you the job of electron transport system is we have accumulated NADH2, we have accumulated FADH2, and now it is time for them to pull the electrons from them, give it to oxygen, in that process to synthesize ATP, and that is said to be electron transport system. And we are going to discuss about electron transport system now. Is it difficult part? Nahi. It is not difficult. Just concentrate here. We'll make it look easy. By this time you know everything, but we'll look at the different ways in which the questions are asked, all right, in this particular topic with regard to this electron transport system. So first, let me draw this electron transport system, children, such that it becomes easier for you to remember all these reactions. All right, electron transport system reactions we'll be writing here. Okay, we'll be writing here. Chalo, first listen here. Concentrate here. So till now you understood everything. So in the last 10 minutes of the class, all right, we'll try to become extremely good in ETS part. I'll go little slow. Right. Mitochondria, I forgot to talk a cell organelle. All right, we are thankless not talking about mitochondria. Mitochondria has 2 membranes. The inner membrane is thrown into number of folds, and this is called cristae. This is the matrix. What happens in matrix? Link reaction, pyruvic acid, acetyl-CoA, and Krebs cycle occur in matrix. And electron transport system will occur on this membrane. Electron transport system is going to occur on this membrane. Which membrane? Cristae, on the inner membrane. Thylakoid membrane, I told you, that is for photosynthesis. Electron transport system occur on the thylakoid membrane. Yesterday we had a marathon class on photosynthesis. Those who have not watched this photosynthesis topic, just manufacture some time to see it. It is extremely handy for you to remember things once for all. You can do the last rites of this chapter. All right. Now, today we'll talk about one more membrane. This is the cristae, the inner membrane. And this inner membrane, all right, is a place where ETS, electron transport system, occur. Yesterday I told you stroma, thylakoid membrane, lumen. I have written like that. Same way I want to write here. Perimitochondrial space, this is called cristae, and this is matrix. I repeat again. Perimitochondrial space, cristae, and matrix. So easy to remember. Shall I write here? I'll write here. This is perimitochondrial space, and this is cristae, and this is matrix. So nice, so nice. I have written nicely. 3 things. How do I write? Why did I write here? This is the perimitochondrial space. This is the cristae, and this is the matrix. Perimitochondrial space, cristae, matrix. Same. Perimitochondrial space, cristae, and matrix. Now what did I do? On the cristae, listen children, on the cristae, I am drawing some enzyme complexes. All right, what are those enzyme complexes on the cristae? Look here, this is on the cristae. So this is the lipid bilayer. So I'm drawing like this. This is the lipid bilayer. Okay? Cristae will have membrane. Membranes are made up of phospholipids. This is the lipid bilayer, which I have drawn here. Now, what we are going to do? We'll take this pen, nice pen, right? On the cristae. All right. This is one complex I want to draw. This is said to be complex 1. Then one more complex I want to draw. This is said to be complex 3. One more complex I want to draw. This is said to be complex 4. And one more complex which you know in photosynthesis we studied. This is complex 5. Let me name this. This is complex 1. This is complex 3. This is complex 4. And this is complex 5. All right. Up to here there is absolutely no problem. These are present on the cristae. Right. Now what do I do here? What are the carriers which are present? So complex 1 contain FMN, flavin mononucleotide, and also contain some iron sulfur proteins, approximately around 6. It contains FMN and iron sulfur proteins. Easy way to remember: 1 flavin mono means 1, flavin mononucleotide. Easy way to remember. And what does complex 3 contain? Complex 3 contain cytochrome B. There is an iron sulfur protein here and cytochrome C1. So it contains cytochrome B, iron sulfur protein and cytochrome C1. No problem there. Then what does complex 4 contain? There is a copper containing protein. There is A1 or A3, and there is again a copper containing protein. By this time you know all these, children, right? This is 1, this is 2, and this is called F0. This is F1. Some of you are thinking naught means something which is round is not F0. You also will get the same mark. All right, this is F1 and this is F0. This is F0 and this is F1. Remember that. There we said CF0, CF1. Here we say F0, F1, also called oxysome. You may ask me why all this. I'll tell you why all this. Did we remove hydrogens from pyruvic acid? Yes. Did we remove hydrogens from acetyl-CoA? Yes. Did we give to NAD and FAD? Yes. Did we produce NADH2 and FADH2? Yes. And from there, our job in another 10 minutes is to produce ATP. My watch is hurrying me up. I want to complete in 10 minutes. All right. In 10 minutes, we'll be seeing how this NADH2 and FADH2 will produce ATP. Children, we are very unhappy. I'll tell you why. In glycolysis, we have produced only 4 ATP, and out of which net gain is only 2. For time being, we'll take 4, because if we still decrease to 2, we'll be extremely unhappy. 4. Link reaction, the number of ATP was 0. Krebs cycle, we are expecting huge number of ATP there. Krebs cycle, for 1 molecule of glucose, we produced only meager 2 ATP, succinyl-CoA to succinic acid. Glycolysis 4, link reaction 0, Krebs cycle 2. 4 plus 0 plus 2. We produced only 6 ATP till now. We have to produce gross 40 ATP. We are in the last step called electron transport system. 40 minus 6, we need to produce 34 ATP here. And mind you, what are the substrates for that? NADH2s and FADH2s. Let us count how many we produced. PGL to 1,3-dipga, 2 molecules each, 2 NADH2, glycolysis, yes. 2 molecules of pyruvic acid to 2 molecules of acetyl-CoA, link reaction, 2 NADH2, yes, 2 plus 2, 4. And in Krebs cycle, for 1 molecule of glucose, this side, this side, this side, this side, 2 NADH2, 2 NADH2, 2 NADH2, this is FADH2, 6. Krebs cycle 6, link reaction 2, glycolysis 2, 2 plus 2 plus 6, 10 NADH2 are there with me. Nice. And how many FADH2? 2 FADH. So we have 10 NADH2 and 2 FADH2. With that, we need to produce ATP here. That is said to be electron transport system. Am I getting a little excited? I'll calm down. All right. Now I'm going to explain you things properly, slowly. All right, such that you can easily follow this. Right. Now, first, we'll take that 2 NADH2 produced in glycolysis also into matrix. So now in the matrix I've got 10, this time I'll tell slowly, 2 NADH2 in glycolysis, 2 NADH2 in link reaction, 6 NADH2 in Krebs cycle. So total 10 NADH2. And how many FADH2 we have? 2. Let us see. Let us start with 1 NADH2 molecule. Out of 10, we'll start with 1 NADH2 molecule. What will happen to this? It is losing its hydrogens. I told you electron transport system means loss of hydrogens. So it is losing its electrons, okay, to FMN, flavin mononucleotide. You can call it as prosthetic group of this enzyme. From FMN, it loses its 2 electrons to iron sulfur protein. From here, the 2 electron goes to ubiquinone. Okay. This is nice. Let me tell you, what is the name of this enzyme? Is this catalyzing the loss of hydrogens from NADH2? Yes. Can I call it as NADH dehydrogenase? Yes. NADH dehydrogenase. Perfect. Now, what this enzyme is doing, it is causing oxidation of NADH2, because it is pulling electrons and protons from it. And it is causing reduction of ubiquinone. Why we say reduction of ubiquinone? Because it's giving 2 electrons to ubiquinone. Addition of electron is called reduction. Does it have FMN? Yes. Does it have iron sulfur proteins? Yes. Now, electrons, just like in photosynthesis, electrons are going downhill. When the electrons go downhill, means from lower redox potential to higher redox potential, the difference of redox potential will be positive. When the difference of redox potential is positive, then naturally the free energy will be negative. Then energy will get liberated. Then you may ask me, if the energy get liberated, what will happen? Very important. Let me put green color because that is something which is important. So in this process, when the energy get liberated, you will see this movement of 4 protons into perimitochondrial space. 4 protons move into this is perimitochondrial space, you remember that. 4 protons into perimitochondrial space. Who is the source of energy? The downhill movement of electrons from NADH to ubiquinone is the source of energy for the movement of 4 protons into perimitochondrial space. That's about complex 1, children. We started with 1 NADH2 and we have seen the 2 electron movement. Electron movement release energy and that will help in the movement of protons. Now let us see, ubiquinone is waiting, all right, for us to look at it and talk about it. So therefore, we'll talk more about ubiquinone now. It is accepting 2 electrons. Which one? Ubiquinone. Now listen. What is ubiquinone? It's a lipid. What is this cristae? Lipid. This is nice. Like dissolves in like. Solutions chapter. Same way. Lipids. The membranes are also made up of lipids. Can ubiquinone move freely? It can move freely. It can move from all sides. Is it deeply embedded in the lipid? Yes. Because this is also a lipid. Cristae means membrane. Membrane is made up of phospholipids. So ubiquinone can easily move. Mobile electron carrier. Now what will happen to ubiquinone? Let us now look. It is accepting 2 electrons. From where? From complex 1. Now listen. For every 2 electrons, ubiquinone is accepting. For every 2 electrons, ubiquinone is accepting 2 protons. From where? From matrix. Look here. 2 protons from matrix. Then naturally, 2 electrons, 2 protons, ubiquinone is going to form ubiquinol. What did you study in chemistry? On on reduction give rise to all. So ubiquinone becomes ubiquinol. It is accepting 2 electrons from here, 2 protons from here, and form ubiquinol. It's a lipid, it can move. It runs here, picks. It runs here, picks 2 protons. Then what will happen to this ubiquinol? Listen here. Ubiquinol again forms ubiquinone. Listen, concentrate here. When can ubiquinol again form ubiquinone? Only when it loses 2 electrons. Only when it loses 2 electrons and also loses some protons. How many protons it loses? 2 protons. But it's not in your book. 2 ubiquinones operate. So therefore, the number of protons will get transported will be 4 protons will get transported. And from here, I'll tell you, from here, B, the 2 electron goes to iron sulfur protein, then to C1, and from C1, it goes to, I forgot to tell you, there is another protein here which is said to be cytochrome C. It goes to cytochrome C. Anyway, this is not that much important. Now we are in ubiquinone. Now concentrate here. I'll start again from ubiquinone. It is accepting 2 electrons. Up to here you understood. It accepts 2 protons from matrix, just like plastoquinone. Okay, you understood. It formed ubiquinol, you understood. Ubiquinol loses 2 electrons to cytochrome B and loses 2 protons to perimitochondrial space. Because 2 quinone cycles operate, 4 protons get transported into perimitochondrial space. That's very easy. From B to iron sulfur protein, the 2 electrons go. From there to C1, from C1, the 2 electron goes to cytochrome C. I think the change of pen is needed. From here, the 2 electron goes to cytochrome C. Cytochrome C. What should I call this entire complex as? Listen, this entire complex. It is oxidizing ubiquinol and reducing cytochrome C. Therefore, this complex is rightly called cytochrome C reductase. Kyun? Because this complex is reducing cytochrome C. Shall I give a simple name? Cytochrome B-C1 complex. It has got an iron sulfur protein. It oxidized ubiquinol. Why oxidize? Because it is removing electrons and reduce cytochrome C. This is complex 3. How many protons get transported? Just like in complex 1, here also 4 protons get transported to perimitochondrial space. And ubiquinone, being a lipid, it can easily move on the cristae, within the cristae. Within the lipid bilayer, it is deeply embedded. It's a lipid molecule. It can easily carry electrons. Children, electrons find very difficult to move within this cristae because they're negatively charged. Charged particles cannot move properly. We already studied in plasma membrane chapter. But ubiquinone says, don't get worried. I'm going to accept electrons. I'm going to accept protons. I'm going to go. I'm going to run. Is ubiquinone doing exactly the same job as plastoquinone, getting reduced on this side and getting oxidized on this side and releasing protons? Yes, reduced and getting oxidized, releasing protons. Complex 1, look here children, when the energy gets liberated, this complex 1 enzyme undergoes some conformational change that creates some channels for the protons to move. But when it comes to quinone cycle, there are no channels. It is oxidation, reduction on one side, oxidation of another side, releasing protons. So understand that that may be useful for your future years, but not for this coming NEET exam. All right, from C1, we are here. C1 to electron goes to C. Now what will happen next? All right, from C, the 2 electron goes to copper, from then to A1, then to A3, then to this copper, and ultimately the 2 electron goes to half molecule of oxygen. What should I call this complex as? See what is happening. It is causing oxidation of dash, cytochrome C, yes. It is causing reduction of dash, oxygen, yes. Can I call this cytochrome C oxidase? Yes. Cytochrome C reductase, because it's giving electrons to cytochrome C and causing its reduction. Cytochrome C oxidase, because it's pulling the 2 electrons from cytochrome C and causing oxidation. NADH dehydrogenase, complex 1. Cytochrome C reductase, complex 3. Cytochrome C oxidase, complex 4. Again, children, when electrons go downhill, energy get liberated. In that process, you know what should happen? Energy get liberated, and now the protons are pushed. All right, now 4 protons are getting ready. Just like here 4 protons, here 4 protons, here also 4 protons are getting ready. But oxygen says, please give me 2 protons. No, because I've got 2 electrons, I want 2 protons such that I can form water. Then out of 4 protons, 2 protons will go to oxygen such that it can form water. So it means that the number of protons transported here will be only 2. Yes or no? Because the 2 protons reach here. 2 protons, 2 electrons, half O2 forms 1 molecule of water. Oxygen. Interesting. In respiration chapter, C6H12O6 plus 6O2. Glycolysis, no address of oxygen. Link reaction, no address of oxygen. Krebs cycle, no address of oxygen. Electron transport system, initial phases, no address of oxygen. You thought for a while that oxygen is never used. Suddenly you see oxygen used. So 1 NADH2 will give ultimately its 2 electrons to oxygen. So if I'm NADH2, I can lose 2 electrons, and there is somebody to take those 2 electrons, and that is oxygen. If you look at oxygen count, children, for 1 NADH2, how many oxygens are utilized? Half. 10 NADH2, 5. Why 10? Glycolysis 2, link reaction 2, Krebs cycle 6. So for 10 NADH2, 5. I repeat again. For 1 NADH2, half. For 10 NADH2, 5. Same way, for 1 FADH2, half. For 2 FADH2, 1. 5 plus 1, 6. That's what we have written the equation. C6H12O6 plus 6O2. Why 6O2? For 1 NADH2, we utilize half molecule. For 10 NADH2, 5. For 1 FADH2, half. For 2 FADH2, 1. 5 plus 1, 6. At last, we have seen 4 plus 4 plus 2. 10 protons in perimitochondrial space. It is acting as a proton reservoir. More protons, less pH, acidic. Protons are moving from low to high. Look here, low to high, less to high. Now what we do? We allow these protons to move back. Your textbook says every 4 proton movement will produce 1 ATP. When they move along the concentration gradient, chemiosmosis, Peter Mitchell. When you break this gradient, first we created gradient. We pumped protons on perimitochondrial space. More protons here. Less protons here. Now we transfer protons from more to less. And in this process, energy is liberated. 10 protons when they move back. 10 protons when they move back, how many ATP synthesized? For textbook says for every 4 protons, 1 ATP. So for 10 protons, 10 by 4 is 2.5 ATP. This is looking absurd. We'll make it round figure, 3 ATP. So therefore, the number of ATP, when these 10 protons move here, all right, then when the 10 protons move here, move back, the number of ATP synthesized is 3. Now it's very clear. This is called oxidative phosphorylation. Oxidative phosphorylation, because you're oxidizing NADH2 and producing ATP, hence it is called oxidative phosphorylation. Now you understood. 1 NADH2 produce 3 ATP. And how many NADH2 are there with us? Let us count. Glycolysis, 2 NADH2. Link reaction, 2 NADH2. Krebs cycle, 6 NADH2. 2 plus 2 plus 6, 10 NADH2. 1 NADH2, 3 ATP. 10 NADH2, 30 ATP. Oh, Huge number we got. All right, 30 ATP molecules we produced, children, in the last 5 minutes. And the first 45 minutes we have produced only 6. Last 5 minutes is very fruitful for us, very encouraging for us, because one NADH2 was able to produce 3 ATP. Like that, we have got 10 NADH2s, 30 ATP. Let us count how many ATP we produced. Glycolysis, 4 by substrate level phosphorylation. How 4? 1,3-dipGA to 3-PGA2, PEP to pyruvic acid 2, 4, finished. Link reaction, 0, bad. Krebs cycle, 2 molecules of succinylchoate to 2 molecules of succinic acid 2. So 4 plus 0 plus 2, 6. In electron transport system, for 10 NADH2, 30. 4 plus 0 plus 2 plus 30. 4 plus 0 is 4, not 0, plus 2, 6. 6 plus 30, 36. But how many ATP we need to synthesize? 40. How many we got? 36. What about the remaining 4? Don't forget, we have 2 molecules of FADH2 also. Yes or no? So now we take 2 molecules of FADH2. That is complex 2. This is complex 2, all of you know. This is succinic dehydrogenase. It is an enzyme of Krebs cycle attached to membrane. We already discussed that. This is complex 1, this is complex 2, this is 3, 4 and 5. Complex 2 is going to oxidize succinic acid and convert it into fumaric acid. This is succinic dehydrogenase. FAD is accepting that hydrogens. FAD is accepting the hydrogen from succinic acid to form FADH2. Look here what will happen. FADH2, just like NADH2 loses to FMN, FADH2 lose its 2 electrons to ubiquinone. And I want to stop here. I want to stand here. Look here, nothing is here. Nothing is there here. Look at FADH2. One FADH2 directly losing 2 electrons to ubiquinone. For every 2 electrons, it picks up protons. 4 protons. All right, 4 protons. 4 protons through complex 3. 2 protons, 4 plus 2, 6. When 6 protons move back, the number of ATP, 4, 1 ATP, 6 means 1.5, so 2. So now you understood, 1 FADH2 will produce 2 ATP. Why only 2 ATP? Look here, this 4 will not be there. Why? Because it is bypassing complex 1, bypassing complex 1. FADH2 goes 2 electrons to ubiquinone, 2 protons, 6. When they move back, 2 ATP. Now you understood, 1 FADH2 produce 2 ATP. Like that, we have got 2 FADH2. The number of ATP produced will be 4. Finished. We got the entire 40. How? Glycolysis. 4 ATP by SLP. Link reaction. 0 ATP by SLP. Krebs cycle. 2 ATP by SLP. Electron transport system. 30 ATP from 10 NADH2. Because each NADH2 produce 3 ATP. And 4 ATP from 2 FADH2, because 1 FADH2, 6 protons means 2 ATPs. Why 2 ATP? 6 protons, when they move back, 4 proton produce 1 ATP, 6 means 1.5 ATP means 2. So 1 FADH2, 2 ATP, 2 FADH2, 4. Now it comes to 40. So this is not bad to understand. You understood everything. All right, let us now look at what I have written with regard to these cycles. That completes this topic. All right, it occurs on Christe. Oxidation of reduced coenzymes. Total 10 NADH2 and 2 FADH2. 6 molecules of oxygen are used. Oxidative phosphorylation. 4 complexes, complex 5 is difficult to tell. Complex 5 is not participating in electron transport. All right, but it is a part of electron transport system. So if they ask you how many complexes are a part of electron transport system, better answer is 5. Even though in the 5th complex, there is no movement of electron. Complexes for oxidation of NADH to 4, 1, 3, 4, 5. Complexes for oxidation of FADH2, 4, 2, 3, 4, 5. All right. Utility of NAD, use the substrate. If this NAD and FAD formed, this is nice cycle. Look here, this is NADH2. Protons get transported. Here 4 protons. Here 4 protons. Here 2 protons. Total 10 protons. And for FADH2, this is not there, no, this is not there for FADH2. So therefore it goes like this only. So 4 plus 2, 6 protons. When they pass through coupling factor, 4 proton produce 1 ATP. So therefore 6 protons means 1.5 ATP, means 2 ATP, provided if it is FADH2. For 10 protons, 3 ATP. This is easy. Complex 1. Last 3 minutes, okay. Complex 1. NADH dehydrogenase, common name. It oxidizes NADH, reduce ubiquinone. Prosthetic group is FMN. It has got iron sulfur proteins. And it NADH oxidation of glycolysis, link reaction and Krebs cycle. Not FADH2. Complex 2. Succinic dehydrogenase, common name. Oxidized succinic acid, reduce ubiquinone. Prosthetic group FAD. It has got an iron sulfur protein. It helps in oxidation of FADH2. Complex 3, cytochrome B C complex or cytochrome C reductase. It oxidizes ubiquinol, reduce cytochrome C, prosthetic group B and C1. There is one iron sulfur protein between B and C1. Function NADH oxidation and FA. For both it is common. Complex 4, cytochrome A1 A3 complex or cytochrome C oxidase. It oxidizes cytochrome C, reduce oxygen. A1 and A3 are prosthetic group, iron sulfur protein 0, copper protein 2, and this helps in both. Complex 5 is ATP synthase, children, you know it. All right, proton translocation along the gradient. F0 is proton channel, F1 is a catalytic site. Is it a part of ETS? Take it as yes. Number of protons needed to produce 1 ATP 4. Number of ATP produced from 1 NADH to 3. Number of ATP from 1 FADH to 2. Balance sheet. Substrate level phosphorylation. Glycolysis 4. Link reaction 0. By substrate I'm talking about. Krebs cycle 2. So total 6. Oxidative phosphorylation. Production of ATP by oxidation. From glycolysis NADH2. This is not glycolysis. Glycolysis NADH2 2. 6. Link reaction NADH2 2. 6. Krebs cycle NADH2 6. It's 18. Krebs cycle FADH2 4. If you add all, 34 ATP. 34 ATP is formed from oxidative phosphorylation. Substrate level phosphorylation 6. Gross is 40. Net is 38. Because we have used 2 in there. So if you separate each one, glycolysis, SLP 4. Oxidative phosphorylation is not a part of glycolysis because the 2 NADH2 are formed in glycolysis. They undergo oxidative phosphorylation 6. So the quota, the total contribution of glycolysis is 10. The total contribution of link reaction, what is this? SLP 0, all right. Then oxidative phosphorylation 6. The total contribution of link reaction is 6. Krebs cycle, SLP is 2. Oxidative phosphorylation 22. Why 22? 6 NADH2. 6 3s are 18. 2 FADH2. 2 2s are 4. 22. So this is the total contribution of Krebs cycle. That's all, children. Amphibolic, I already told you. Glycerol enter at PGAL. Fatty acids enter at acetyl-CoA. Amino acids enter at any points. These are not only entry points but also exit points. That is the reason why it is amphibolic. And I did not want to talk about RQ value. For carbohydrates it is 1. For fats it is 0.7. For proteins it is 0.9. And for organic acids more than 1. Organic acids are rich in oxygen. Therefore they require less amount of oxygen. So more CO2 by less oxygen. Therefore RQ value is more than 1. Fats and proteins are poorer in oxygen. When they are poorer in oxygen, they require more oxygen for oxidation. So less CO2 by more oxygen, then naturally it will be less than 1, 0.7 for fats and 0.9 for this. That's all. That's about respiration. I think you are not getting suffocated anymore. This topic looks more easier now. Thank you. We'll discuss with plant growth in tomorrow's class.